Yo, welcome back to the NucleoGenesis project. My name is Dr. James Utley. I'm your resident codebreaker data scraper. I'm a board certified longevity scientist as well as a certified advanced biotherapies professional. I have a PhD in health science, and here we are. NucleoGenesis is a project out of Syndicate Laboratories, and NucleoGenesis specifically is a temporal framework for eukaryotic nuclear blueprint archival and reconstruction. So, taking the information from testing, building a nuclear blueprint manifest, taking that manifest, hopefully generating a synthetic nucleus in the future, amongst other things. And so we are on part 12. Part 12 already. Part 12 puts us at part 6. Part 12 of the video series puts us at part 6, which is discussion, which is the synthetic gap and future directions, right? So now we're on the now we're done with the unveiling the project. So let's just take you back real quick. First one is we talked about the nucleosampler, which is the ability to take a fresh sample, test it, archive the information or collect the information, and then also inoculate a dried blood spot card, keep that in a preservation state, cryptographically logging every step. Now you take that. Now you're going to build a digital file, a digital twin, which is a nuclear blueprint manifest, right? So that's through the Nucleo Builder application, and it goes through building it with confidence tiers, taking all the information we know, providing some statistical imputation to build fill in the gaps, and then build a data anchor layer. That's both fresh sample and well as long horizon testing, right? So then we go to nucleo reconstruction, which is the 3D modeling, which is for the user to 3D model to see how dense the chromatin is, and what the nucleus looks like on a 3D model wrapped in some cytoplasm on an axis. So then we move to the nucleosynthesis, right, which is then how do you add more information from the future into the nucleogenesis framework, and all while keeping all the information that you have in terms of the nucleo blueprint manifest and everything that you've done in between, all version controlled, all in immutable ledger. And then so that brings us to the last part, which is the gaps, So this is a theoretical framework, obviously, right? And there's things that are, can be conducted now. But of course, this framework exists in actually building a nuclear blueprint manifest that can be then synthesized in the future. It is dependent on filling in some of these gaps. That's why we have a collect now, decrypt later kind of overarching theme. So here we are. So first, we're going to start out. What does nucleogenesis deliver today, right? And so nucleogenesis, as a framework, delivers the most comprehensive in silico representation of the human nucleus as a physically, biologically, and epistematically achievable with current experimental and computational technology. It is important that it doesn't just represent a speculative reconstruction, but it has a constraint and the synthesis, which is around measured data, right? So it's a bound inference, it's measured data, and with the uncertainty layer, it's an uncertainty aware model. Okay, so we break it down. So what does it deliver today? Like what can we actually do? We can actually do these tiers. So the first one is a complete genome sequence with phased variants. Like that's what we can conduct that today. Nucleogenesis produces a reconstruction grade linear genome sequence derived directly from the donor material, including the single nucleotide variants, small indels, the structural variants, the haplotype phasing, and that is all supported by the data because this is testing that we can do today, which is a whole genome sequence. Now, another thing that we can also do is the methylome profile and that is stable across decades, right? So the genome-wide methylation patterns are recovered from fresh samples and the dried blood spot material and encoded in a chemically stable epigenetic constraint. Because these marks persist under long-term, dehydrated or desiccated storage, the methylome functions at a time interval, right, provides a good baseline for the next phases or delayed reconstruction and comparative analysis across long horizon decades. Okay, so chromatin architecture from fresh sample or reference based imputation. So when fresh material is available, nucleogenesis captures native 3D chromatin organization through Hi-C measurements. So that can be done today, but that is only done on fresh data, on fresh sample, because the data has to be fresh, right? So you have to also use the population reference atlases that are available for today. But you're gonna have, by doing that, collecting the data and adjusting, you're providing a statistically grounded architectural, which would be a prior, which is a framework. Okay, so 3D dimensional nuclear reconstruction with per locus or loci uncertainty quantification using what we had talked about in prior videos, that it really seeks to quantify the spatial uncertainty around genomic locus, around the genomic locus, and yielding just some predictive values, right? And geometry. Okay. Future-proofed biological archive for iterative analysis. So because we're collecting and impregnating or inoculating in a dried blood spot card, all reconstructions are anchored by the durability of the dried blood spot archive. And because there's software pieces that are important for the version control, that's how you kind of build the framework that just builds and builds and builds on each other, right? So now, so that was the gap, the synthetic gap in the future directions. Now we're going to talk about the digital to physical, right? So despite the theoretical completeness of the nuclear blueprint manifest and the 3D nuclear model and all of these all of these fundamental things that are part of this framework, right? It does not translate to any functional physical nucleus at this time, right? So we know that we've been saying that in all the prior videos. We just know that. So the gap, but we know that the gap is there. But what we can do is to try to provide enough data so we can generate this nucleus, right? So that includes like the sequence, the epigenetic constraints, and all the spatial organization, which is people are different people at different laboratories are working on this, but it does lack the means to really build a physical nucleus. So I think there's a good graphic on the next page here. Okay, great. Yeah, the bridging the digital physical gap in synthetic biology, which is something that I as a data nerd myself love to provide as much data that has this translating context into an actual physical nucleus. Like that would be amazing. But let's talk about where the gaps exist, right? So we have this whole process, right? So we build the nuclear blueprint manifest, and we're trying to translate it to a physical nucleus. Like that's one way. That's aside from the other use cases that we'll talk about in the following chat or the following sections. But we know that cell-free assembly. All right, we can't do that. We can't take information, right? Chromatin, the chromatin organization, right? So that part right there. So we're limited there. Obviously, we're limited there again. DNA synthesis, right? So we're limited in that it's not going to translate to a functional cell, right? So the most immediate so genome scale DNA synthesis, the most immediate and quantifiable limitation lies in DNA synthesis, while digital genomes be can be specified precisely. The physical realization requires the de novo synthesis, assembly, and validation of contiguous DNA at the gigabase scale. So, get giga with it. And then at the present state of the art, our whole genome sequence construct just doesn't, it doesn't, it's not mature enough. I mean, we haven't had a breakthrough that would allow us to do that, right? So, but that's the whole point. It's a collect now, decrypt later, right? So and at the minimum in this application, if you can't ever see a future of a physical nucleus from this nuclear blueprint manifest or the data plan, then what you know is there'll be insights that will then be able to train further artificial intelligence to then be able to bridge that gap. I feel very positive about that. And but here, programmatic chromatin assembly, even assuming that the availability of error corrected gigabase scale synthetic DNA, the construction of a functional nucleus requires far more than linear sequence realization. Like I get that, and I put that in the paper here, so so the reader understands that this is a framework that will still need to be matured as technology increases. DNA must be organized into chromatin in a highly specific multi-scale manner that encodes the regulatory state, the spatial organization, and the dynamic responsiveness. And I think that's going to be really hard, but I believe that as we collectively learn more and we are orienting or pointing artificial intelligence at biological processes, we will get there. We will provide this like a process of this routine in living cells, which we'll understand more fundamentally than we do now. And that's the whole point. Okay, so we have to know that we have we have to know and recognize that correct nucleosome positioning is important. And we have to also know that we have to have ability for the appropriate histone variant and how to incorporate those variants and post-translational modification patterns and high order folding into domains and compartments. Like we get, like you see how it's like some really complicated stuff, and like in isolation we can work on all of these individual things, right? But the point of the nuclear blueprint manifest is to provide enough information in a version controlled way that one day it would be able to answer all these questions with the right imputation, with the right computation of an AI, of the AI models that are far more advanced than humans. All right. Now, yeah, this is one thing we haven't talked about is the nuclear envelope and pore complexes. So the nuclear envelope represents a defining architectural feature of eukaryotic cells, right? We know that, the nuclear envelope, right? So it establishes a physical and functional boundary between the genome and the cytoplasm, right? That nucleoskeleton system, or that linking, right, of the nucleocytoplasm skeleton system is important. So unlike plasma membranes, which primarily serve as selective barriers, the nuclear envelope is an active participant in genome organization, gene regulation, and mechanotransduction, right? So that is an important. So the envelope consists of lipid bilayers to make this double membrane architecture, and the nuclear pore complexes, or the NPCs, are among the largest macromolecules assemblies in the cell. And really being able to correct for those is going to be a key in actually building a synthetic nucleus, right? So here we go more over it, right? And we understand that we need to, you know, that beyond the chromatin organization, which a lot of this project is really focused on, the nucleus is capable of transplantation and functional integration into a cellular environment that must be capsulated with the fully formed nuclear envelope, like we get it. And so the double, as I mentioned, all of these things, the nuclear lamina, the inner nuclear membrane is reinforced by the filament network of lamins, right? Primarily the lamina A and C and lamina B and these different isoforms. And if you remember in a few videos prior to, we have our energy or our E model, which does the backbone, and then it also has the repelling factors, right? So this would be one of them. So the fibers, they can't be like overlapping, and that would be in the nucleo reconstruction model. They can't be overlapping, so they have to have this like digital spatial repelling mechanism. Okay, so active import and export machinery. Functional nuclei requires a complete GTP dependent transport system, including importans, exportins, and these regulatory cofactors. And so these are all, this is, the nucleus of a cell is a very, very complicated space and a lot of things going on. And I would say that we don't understand, meaning like collectively we understand like what everything does and how it works, including myself. I learn something new all the time. But as we go through this, right, we know that that the nuclear blueprint manifest and future technologies and collecting this data in a version controlled, cryptographic way where the data is the important future, or the important feature is that it will hopefully bridge the technologies that are under development. So for example, if you have people working on the virtual cell or digital twins and things like that, like this data can be very useful. It could be very useful because it's collected with the purpose of trying to synthesize a nucleus in the future. And so that's where this comes from. Okay, moving on is really it's just highlighting this graphic of like the cell-free nuclear assembly that they would need to have that worked out. And then you're talking about the nuclear envelope and in between, meaning like the the trans mechanisms that are in between the cytoplasm and the nucleus. Okay, but here's what I wanted to get to for this video was the application horizon. So, as I mentioned before, that it's like the so what, right? So these are the different applications that are kind of the baseline applications that could be used, right? So personal genomic medicine, like if we're using the nuclear blueprint manifest, there's going to be information there that you could derive to conduct whatever personalized medicine strategy that you're trying to implement. And then induced pluripotent stem cell derived guidance, right? So because it's you're working with nuclear material, of course, then There is items that if you're trying to induce a stem cell, you could you could gain some information from that. Forensic identification. So of course that's just taking the information from the nuclear blueprint manifest or the data, the nucleus data, and then you could obviously use it in a forensic application. Organoid engineering. So maybe the architecture or the nuclear blueprint manifest, the NucleoGenesis framework could inform organoid engineering, especially for yourself, right? Like it could be the information could inform that, especially if science is, meaning like if that field is advancing, it could help. And then, but the synthetic chromosome construction, it's going to be definitely a different beast, and I think there's a lot of work to do. But then here would be the granddaddy of them all, which is the complete nuclear fabrication, which is making a physical nucleus in the world from a digital file, which would be amazing. And so, where would you use it? Okay, so disaster resilient genomic backup. That's obviously right in front of your face, right? So if something bad happens in the world, which bad things are happening all the time, and even more so today, that you would have, unlike in cryobanks, which depend on continuous energy input and institutional stability, and the dried blood spot archives would be very, very useful because they could be, you could reconstruct or you could use that information. And this is the foundation for biological continuity, right? So deep space support. So if we're talking about organoid engineering or print me a new organ or anything like this, or even just help induce pluripotent stem cells, then a deep space mission. So in extraterrestrial or long duration space flight context, fresh biological sampling and cold chain preservation is just not feasible. So dried blood spot analysis, storage, and transport of genomic and epigenomic information with minimal mass, volume, and energy requirements. As a result, a DBS provides a practical framework for genomic medicine, personalized risk assessment, and biological monitoring in environments where traditional laboratory infrastructure cannot exist. So let's get the real crazy pie in the sky is that you would send you would send your DBS card that has been collected and your digital nuclear blueprint manifest. You would send it with the next space flight to Mars. They would go ahead and then print you another body, and then so all you have to do, or let's say it's farther, it's got to be farther than Mars, but let's just say, or I guess so. And then all you have to do is preserve your consciousness through your brain, and then when you get to the new place, you can get a new body. So intergenerational genomic records, and again, and then future proofing for reconstruction. So those are the whole thing. I challenge you to go read this paper. It's pretty amazing. I like it. It's good. It's some years of work, and some of it's disjointed, but some of it flows together nicely. Okay. Okay, this is part 7, which is the conclusion. So this will be the last. So this is part 12, and then I'll come back with part 13. That's a great number to land on. And again, this is NucleoGenesis project found at syndicate-labs.io, syndicate-labs.io. We're building a nuclear blueprint, and which is a nucleus to a cell. And again, my name is Dr. James Utley, and I will see you in the future. Peace.